What you see here on the screen is a detail of what happens with photosynthesis. The first thing you need to notice is that here we have the equation for photosynthesis. On the left side of the arrow are the reactants, and on the right side are the products. Make sure that you write that down completely and clearly. 6H2O plus 6CO2 plus light yields C6H12O6, which is glucose, and 6O2. There we go. You can see that a little better. Remember that the word photosynthesis means to make with light. Photo means light. Synthesis means to make. We start with the sun, which are little packets called photons. That's light. And H2O entering into the chloroplast. You notice this structure that we see here on the whole diagram is the chloroplast, which is inside a plant cell. The photons come in highly energized and break apart the water molecule in a section called the light-dependent reaction. This happens in these stacks of grana, and each one of those individual stacks looks like plates is called a thylakoid right there. The electron transport chain, right here, ETC, happens inside the light-dependent reaction, and it is a highly, highly volatile reaction. You don't have to know everything that happens in this reaction except for the H2O is broken apart to where the oxygen is spit out, and it is a waste, but the hydrogen attaches itself to the NADP. This becomes a highly energized molecule. Remember that a hydrogen atom has only one proton and one electron. So the proton attaches itself to NADP, highly energized. The electron gets spit out by itself, and the NADPH, the electron, and an ATP that's just sitting around here waiting, all enter into the Calvin cycle. This had to happen because the photon had to energize the hydrogen. That is what actually happened in order for this reaction to take place. It enters into the Calvin cycle, which is out in the stroma, which are the spaces in between the grana. That's where the Calvin cycle takes place. It is light independent. That means that you do not have to have light because the energy from the photon is already here in these three molecules. We don't have to know exactly what happens in the Calvin cycle, but you do need to know that CO2 comes into the Calvin cycle. It becomes highly energized, and all that energy gets packed into a glucose molecule that we see here, C6H12O6. So basically, the energy from the photons of light gets transformed into some high energy molecules that then remake it themselves into glucose. ADP, which is an adenosine diphosphate, comes back around to be re-energized, as does the NADP. Not so as important. Things you need to take away. The reactants, which are at the top side, the sun, the water, the CO2, enter into the chloroplast. The water specifically goes into the thylakoids or the grana for the light-dependent reaction. The CO2 goes into the stroma for the Calvin cycle or light-independent reaction. The products, O2, which is considered a waste at this point, and C6H12O6. Again, our reactants are up here. Our products are down here. I wrote some things on the side, ETC, electron transport chain, ATP, adenosine triphosphate. Everything that's highlighted in orange, there actually should be one more thing, the stroma. These are locations where things happen. Everything else is not a location. It's just either the name of the reaction or it's a name of a molecule. All right, so the first thing we want to do is prepare this. We're just going to do some writing on it right now to get it ready for us to work on. So if you'll go ahead and write photosynthesis here. I'm sorry, say that again. Sideways? Yes, we're going to write on it sideways. Wow, okay, yeah. Thought I was spelling it wrong all of a sudden. So on one tab, write photosynthesis. On the other tab, write cellular respiration. I will tell you this, that this particular foldable is going to be the key to you understanding photosynthesis and cellular respiration and their relationship with one another. When we get ready for the test next week, when you're studying, if you know what's on this foldable, you are going to be good to go. Okay, so turn it kind of sideways like this. On this first tab, not the one that has photosynthesis and cellular respiration, but the first tab right here, write where, and then reactants, Are reactants on the left or right side of an equation? Left. That is right, Austin, the left. So the products are on the right. Then write purpose. And then across the whole bottom, we're going to write chemical formulas. Oh, that's not even on the screen all the way. So I think that one of the best study tips, and I did this, y'all know I just finished my master's and I did this a lot, is drawing pictures. Pictures help you visualize what you want to look at. So photosynthesis, what type of organisms undergo photosynthesis? Absolutely plants. So let's be a little artistic here. I'm going to draw me some grass. Yeah, this is my grass, unmowed. Looks like my front yard. Y'all can draw your grass however you want to. I'm going to draw me a little tree coming up through my photosynthesis word. Kind of coming out. Oh, it's a big tree. Just draw, peeps. Just draw. Okay, I'm drawing me a big tree. That's a good climbing tree is what that is right there. I'm going to draw me a little flower, a little wildflower over here. So just a visual reminder of what photosynthesis is. All right. Once you've done that, flip over to the Where tab, the next tab. So what is the name of the organelle where photosynthesis takes place? We just talked about it a minute ago. It is a chloroplast. CHLOROPLAST. And last time we did the diagram that had the chloroplast. It's kind of an oblong, oval shaped organelle. And inside are these stacks. You remember we talked about the stacks? They kind of look like stacks of plates, stacks of coins. Money, money, money. I'm not a very good artist, so just go with it. That's what it looks like. And these stacks are called grana, G-R-A-N-A. My G kind of... The stack is called a grana. We talked about this last time. The individual little coins, if you will, if that was a coin or a plate, are called the thylakoid. And the spaces in between are called the stroma. If you can't remember the spelling, they're written on your page, your glossary. Stroma, S-T-R-O-M-A, stroma. Okay, so the reactants for photosynthesis, there's 3 of them. Look back at your page from last time. What are they? Reactants. On the left side. Look on the left. Water, yes. Water's one of them. Carbon what? Carbon dioxide and sunlight. Again, I like to draw little drawings. For water, I don't know, y'all can draw whatever you want. I've been drawing little droplets to help me remember water. The actual molecular structure of water is something like this. There is an oxygen with 2 hydrogen on one side. It's a polar molecule. It actually almost looks like Mickey Mouse. When I think carbon dioxide, I think soda or a Coke. So I'm going to draw me a Coke can. Did y'all know that's what makes the Coke kind of fuzzy, fizzy? Say, carbon dioxide in it? Yeah. Okay. The actual molecular structure of carbon dioxide is more like this, with a carbon in the middle and 2 oxygens sticking out on either side. And then the sunlight, of course, we're going to, something you've been drawing since you were in kindergarten. We're going to draw the sun. The products. So if reactants are on the left, products must be on the right. Okay. Look at your illustration for photosynthesis last time. What are our 2 products? Glucose and oxygen. Oxygen is actually considered a waste at this point, although it will be used in cellular respiration here in a minute. Oxygen, I have no idea how to draw it. I just always draw these little wispy things. I don't know. Y'all can draw if y'all are good. Yeah. If y'all are good artistes, maybe you could draw an oxygen mask or, something a little bit better than my representation. And then for glucose, I like to draw a little sugar package to remind me that glucose is a sugar. Okay, so what is the purpose of photosynthesis? Does the plant do this for us? No. Why is the plant doing this? For it. What's it making for it? Food. So the purpose, we're on the purpose tab, is to create food for the plant using energy, again, that, non-matter energy, little photons from the sun. If it wasn't for these plants, you'll need to understand, like we talked about in ecology at the beginning, there would be no life on Earth. Period. End of story. They have to do this for the world to survive. So from the beginning of life on Earth, it started here. No animals could have survived if there weren't plants to eat. Get it?
Wait, so technically, the first ever born animal was
Well, it had to be in order for any life to start, it had to start with autotrophs, things that could make their own food, right? Then herbivores came along because they eat what? Plants, okay? So, yes, is that what you were going for? Okay. All right, so let's go to the formula. You may want to wait and let me write it so you can kind of get your spacing down. It is 6 CO2 plus 6 H2O, which is what? Water. Plus light. It is. It doesn't change. But here's the deal. By you rewriting it, you get it. So it yields glucose plus 6 O2, oxygen. Cellular respiration. I'm going to focus in on that a little bit because that is hard to see. It is 6O2 plus C6H12O6 yields, and I'm sorry that bubble's getting in the way a little bit, 6CO2 plus 6H2O plus ATP. You'll notice it looks really similar to what? The other one, which was photosynthesis. That's intentional. That's a big thing. Okay? All right, so we're going to get our color coding situated here. Like I told you, you need 4 colors. Anything I'm using highlighters. You can use your color pencils. Do y'all see right here on the right where it says key? Yep, okay. So I'm going to use my colors that I've chosen. Of course, yours aren't going to be the same necessarily, and that's okay. For my carbons, I'm going to use pink. And the reason y'all can just highlight straight through, but when I do it on the screen, it ends up looking blacked out. So I'm just going to circle them. My locations I will do in blue. Every time I see oxygen, I'm going to highlight that in green. And then anytime I'm talking about ATPs, I'm going to do that in yellow, which it looks quite fluorescent up there. So you're just going to need to pay attention to your own code as we go through, because like I say, yours isn't like mine necessarily. All right. Couple of things I want you to notice. We are dealing with a cell here, okay? So everything that is inside this circle is inside of the cell. What do you notice right over here? It's called cellular respiration. What do you notice this word is right here? Can y'all see it? Aerobic. Look on your glossary. What does aerobic mean? Something? Requires oxygen. Have you ever done aerobic exercise? What do you start doing when you do aerobic exercise? Breathing a lot, right? Because it requires oxygen. Question?
Why did you do it that highlighter?
I don't know. Should I have done it in green?
Yes.
Oh, okay. So sorry. Okay, because it does require oxygen. Thank you. That's right. Okay, for my carbons, right here is an important note. Carbon bonds do what? Hold the energy, okay? So throughout class today, we're going to be tracking how many carbons each of these molecules have, because it is so very important as to how much energy there is, because the bonds hold the energy. All right, so let's start the story. Let me back out here a little bit with glucose. Glucose, how do you think we get glucose? How do I get glucose into my body? Through doing what? Eating food, right? So the glucose comes into my body through my food source. Actually, there's a little bit of breaking down that your stomach and intestines will do until you get it into this glucose format here, C6H12O6. Now, glucose is a huge molecule. Let's go ahead and highlight that with our our carbon color. Glucose is a huge molecule, and it cannot get through the cell membrane by itself. It has to have a specialized hormone called insulin. Where have you heard of insulin? Diabetes, right? Okay. Diabetics do not have the ability to produce either enough insulin or any insulin at all. If you're type 1, you probably are not making any insulin at all because something was wrong with your pancreas from the beginning. If you're type 2, that means you develop insulin resistance or diabetes over a lifetime. Insulin's job is to take this glucose molecule from outside of the cell and push it into the cell. So if you are diabetic, that means that glucose molecule is not getting inside the cell ever. And so guess what? What did we say the glucose molecule or the carbons are responsible for? Energy. And if you're not getting that glucose molecule inside your cell, you're not getting energy. Have you ever seen a diabetic when they're going into what they call low blood sugar? How do they behave? Oh, that's right. Very tired, very lethargic. They might get kind of shaky. They might even get a little bit grumpy, right? Oh, yeah. Right? Because their body is basically in shutdown mode, and their body is saying, get energy now. So their personality kind of takes a back seat to their need for energy at that moment. So diabetics will take insulin in order to push this glucose molecule into their cell. If they don't do that, sugar outside of the cell will start damaging tissues. They'll start having damage in their fingertips. They'll start having damage in their toes, even eventually in their kidneys. And so they have to have that glucose molecule pushed in there. So either they'll take artificial insulin, or sometimes they have a pump. Have you all ever seen like a type 1 that has a pump that's distributing their insulin all day long? That's a really good method for type 1 diabetics, is to have that pump. So that, I want y'all to kind of have that in mind as we go through cellular respiration, why that's so important. We just had lunch. Even people who are non-diabetic can kind of feel that way. Right before lunch, don't you start feeling a little bit lethargic, kind of tired? You need a boost of energy, right? And the reason is because your body has used up all this glucose, and so you're out of ATPs. ATPs don't get stored. ATPs are used minute by minute by minute. And so you have to be refueling that production of ATPs, which is cellular respiration. Have to. No choice. Okay, so the cell membrane, that's what this line is represented by. The glucose is pushed through into the cytoplasm. These are locations, if you want to mark those with your location color. Glucose is a highly stable molecule. It will sit like this forever. It'll sit on the shelf and never do anything. So we have to create a little bit of instability. And the way we do that is by adding in some ATPs. So these ATPs here are used. Do you see how their arrow is going into the reaction, not away from? So they're being used, and they immediately create these little molecules here called PGALs. We don't need to know what PGAL stands for. The big important thing you need to know here is it is unstable. What do you know about unstable molecules? What do they want to do? Move, blow up, get going, all right? So these guys are highly, highly, highly unstable. That is good. This is the beginning of this process we see right here. Let's kind of draw a line right here. What does glyco mean? What do you think that's referring to? Which sugar? Glucose, that's right. And what does lysis mean? We've talked about it with lysosome. What does lyse mean? What do our lysosomes do? They break things apart, right? So lyse means to break. So we have started the process by adding these ATPs of breaking apart the glucose. And the reason is glucose can get into the cell, but it cannot get into the mitochondria. What happens in the mitochondria? What have we learned about the function of the mitochondria? It is energy. It is powerhouse. So what we're trying to do here is break this glucose down and get it small enough to where it can get into the mitochondria. And that process is called glycolysis, which happens out here in the cytoplasm. Well, the first step was to add 2 ATP, and therefore 2 PGALs are formed, which are, important thing, unstable. So 3 things happen to the PGALs. They are so unstable that immediately 4 ATPs are pushed out. Right away, boom, they're made from the breaking apart. Every time we break these carbons apart, every time a carbon comes apart, energy is let out. So that carbon was broken apart a little bit and energy was let out in the form of ATP. A hydrogen was let out, or actually 2 hydrogens were let out, and they get aboard this NAD, kind of the same thing as in photosynthesis, and they enter into an electron transport chain, ETC. We talked about electron transport chains last time, and I called them, and I want you to write it over here. Anytime we talk about electron transport chain, I want you to think about the mosh pit of energy. There is massive chemical reactions happening in an electron transport chain. It is just going with energy all the time. So much so that we actually end up producing 6 ATPs in this electron transport chain. Look up here at the top of the electron transport chain. What molecule is entering into the electron transport chain? It's tiny. You'll see it right there? O2, oxygen. Okay, let's go back to, I'm going to circle it with my oxygen color. What was the name of this process over here? Cellular what? Cellular respiration. What does what say where? All right, so we see here that oxygen's driving the electron transport chain. What's being given off of the electron transport chain is a waste. H2O. Look over here at the reactant side. Bam. H2O, right? Okay, so I told you when the PGAL, the unstable PGAL, breaks apart, 3 things happen. 4 ATP, our first electron transport chain, and the third thing is that glucose molecule is being broken down into 2 pyruvic acids. Tracking our carbons, how many does each pyruvic acid have? 3. It's getting smaller, right? Instead of a 6-carbon molecule, it's now a 3-carbon molecule. In the video we'll watch in a minute, they'll call it pyruvate, but it's the same thing, pyruvic acid. How many pyruvic acids are there? 2. Do you see how I wrote it twice? So that means from here on out, everything that happens past this pyruvic acid happens twice for each glucose molecule. So you might want to write times 2. What are we trying to get rid of to make it smaller? Carbons, right? Well, right here, a waste product of CO2 is given off. Do y'all see it? Look over here, CO2, okay? So the CO2 is given off. When that carbon CO2 breaks off, it immediately causes another electron transport chain. So we'll highlight him. And we end up with 3 ATPs. Bam. And again, what molecule is driving that? What's going in to make that happen? Our magical oxygen. You see, there's a lot of stuff that actually happens in these electron transport chains. The video we'll watch in a minute kind of goes through some of them. But what I want you to be aware of is that it's just a huge buildup of energy, making of energy. We're not going to worry about the individual steps that happen in those. Okay, so if I take a 3-carbon molecule and take one of the carbons off in the form of CO2, how many carbons do I have left? 3 minus 1 is 2. So I'm down to an acetyl group, which has 2 carbons in it. It is small enough to get into the mitochondria, but it's a big molecule. It can't go through with passive transport. What do we call it where we use energy to get in? Yeah, we use ATP. Well, what kind of transport is that where we use ATP to get into a membrane? Active transport. So right here we're using an ATP, and this is active transport. It's going to push that acetyl group through into, yay! We're finally inside of the mitochondria. So here's the acetyl group. I basically just rewrote it. And how many carbons does it have? 2. There is a series of reactions that happens here. I am not very concerned that you know what they are. Just that, remember, where are the bonds for energy? Inside the what kind of bonds? Carbon. So we can't get very much energy out of a 2-carbon molecule. So a series of reactions happens here inside this called the Krebs cycle, which is the reaction that takes place in the mitochondria, building back up the number of carbons. There's some carbons sitting around waiting in here for this to happen. And they get built back up until we get to another 6-carbon molecule. It's not glucose. It's actually called citric acid. So it's a chain reaction, basically, to where we build back up to a citric acid that has 6 carbons. What do you notice about it? What's that say right there? Unstable. Again, unstable leads to breaking apart. So those 6, even though we just built it up to 6 carbon, those carbon molecules are going to start breaking off. And the first time they do that, we get another electron transport chain right here. And our biggest production of energy at one time, 9 ATPs. 9. And again, what molecule's driving it? Oxygen, because it's an aerobic process. Instability leads to 1 ATP being kicked off by itself, right there. And then our last little struggle of the Krebs cycle, getting to the end of our energy, kind of like the train getting to the bottom of the hill. It has a little bit left, but not much. And we make 2 ATPs, just 2. And oxygen, again, driving the process. Okay. Very complex, as you can see. And like I told you, I've left off some stuff here. But you can see what molecule is going and making this Krebs cycle keep going. What is this molecule? H2O is water. What molecule is being given off as this? Remember, this 6 carbons being broken apart. So what molecule is coming off? Carbon dioxide, CO2, right? As we break those molecules apart, that carbon's coming off. And every time we break them apart, we're getting energy out of it. This whole process is pretty complex. Here's your big takeaways that I want you to have from this. And that is, what are we doing in glycolysis to our carbon bonds? Look at the pink. Breaking it down so that it can do what? Get into the mitochondria. Once it's in the mitochondria, what do we build back up? Carbons. Once we get it there to an unstable molecule, it breaks apart and creates more energy, right? Okay, so let's do a count of how many ATPs we made. These are on my page, the yellow. So right here is 4, and right here, 6. Do y'all see where I got those two? Okay, now I'm at the pyruvic acid. So everything after pyruvic acid happens how many times? Twice. So going here, here's 3 ATPs, but how many times does that actually happen? 2 times. So that's 6, right? I'm going to write it down twice so y'all can see it. All right, now I'm into the mitochondria and the Krebs cycle. How many times does that 9 there happen? 1, 2. Remember, because we're after the pyruvic acid. So that happens twice. How many times does this 1 ATP happen? Twice. And how many times does that 2 ATP happen? Twice. Okay? Write those down and add them up. See what you get. It's what? 40. It is 40. So we got 40 ATPs. But as any good businessman knows, you don't just get what you get. You had to spend some, right? So how many did I spend to get there? Let's go back to the beginning. Used. So minus 2, right? Going through, where's the next time I used some? Right there. And how many is that actually? There's 1 ATP used for the 1 pyruvic acid, but the other pyruvic acid, so that's 2. So how many ATPs? 40? I'm off the screen, sorry. 40 minus 2 actually comes out to be 36 ATP. Now what I want to point out about this is for a individual, organism, human, dog, rubber tree plant, whatever it is, that is very healthy and working at its prime. We don't always get 36 ATP. And organisms that's going slower or maybe getting older may not get 36, but that's the goal, 36 to 38, because sometimes we get additional ones spit out here and here. But usually it's around 36 to 38, okay? All right, if you'll go back to your foldable over cellular respiration, right here, we're going to, now that we've kind of looked at it in very, very great detail, now we're going to look at it more general. So the first thing, again, I like to use illustrations to help me see, what kind of organisms. So let's draw, what kind of organisms undergo cellular respiration? All of them. So you mean even plants do cellular respiration? They absolutely do. What would be the use of them making glucose if they weren't going to burn it and use it, right? So this is the process they do that. So I'm going to draw me a tree again to remind myself that okay, contrary to what y'all might believe, that is kind of a rabbit. It's some small mammal, okay? Hey, I didn't laugh at your, do you see me laughing at your drawings? Don't laugh at mine. Okay. So I drew all that to remind myself that cellular respiration takes place in both plants, animals. Hey, you know what I should also draw? I'll kind of draw it here. Well, an owl's an animal, but I can't draw an owl. Look how bad my bunny is. Forget it. That, thank you. You know why it looks like a mushroom? Because it is a mushroom. Because fungus also do this. So, good.
It is a mushroom.
It is a mushroom. Okay, so now let's look at where. So you see right here, I'm going to flip up this tab where. All right. What is the name of where most of this energy is made? What's the name of that organelle? No, not the organelle that we're trying to get into. Mitochondria. So we're going to label mitochondria. And the mitochondria looks a little bit like a peanut. And it has this membrane folded all inside of it. Looks kind of like that. I'm a really bad drawer. The actual membrane that's folded up inside is called a cristae. And the spaces in between are called a matrix. I know, not the movie, which I've never seen, and thank you for, yeah, I know.
I've never seen it either.
I need to watch it.
What?
Okay.
You just officially made my papers.
Okay, so it takes place in the mitochondria. Where? It says Cristae, Cristae, and right here it says matrix. Okay, we're now going to go to the next tab, so make sure you're lifting up the correct tab. We're going to reactants. Left or right side of the equation? Left. Always the left. Everybody got that?
No.
You got to stop talking and start writing.
I was helping out someone.
Okay. Let's move to the reactants tab. So our two reactants are oxygen and glucose. Okay. Again, I don't know how to draw these very well, so we're going to draw the little wispies. Okay. And then glucose, I'm drawing a sugar bag. Y'all can draw whatever you want that represents sugar to you. Just a visual of it. Yes, I should be a professional artist. You're right. Okay. I'm now going to look at products. Y'all ready? Products. Go to products. 3 products. H2O, CO2, and ATP. H2O, again, will represent with some little water droplets. I'm going to draw the oxygen with the 2 hydrogens just to remind me. It looks like Mickey Mouse, and that was bad, but okay. I'm going to draw the soda. Soda. Coke bottle. Again, the carbon in the middle with 2 oxygens on the side. And then my energy star of ATP. Something kind of out of the cartoons. I'm not very good at it. Somebody last period said, can we draw an energy bolt or a lightning bolt? Sure. And then I want you to go ahead and write out the word so you'll have it written down here. Adenosine triphosphate. Adenosine triphosphate. And I'm enlarging it on the board so you can see it even better. There you go. So how many phosphates are in adenosine triphosphate? Tri. 3. That's right. 3. Okay, look up at me so I'll know you're ready for purpose. Okay, let's turn to purpose now. What is the reason we do cellular respiration? What are we trying to get? Energy in the form of what molecule? ATP. So we're going to use food provided by the plant to create usable energy in the form of ATP. That's a pretty concise statement. Use the food provided by the plant to create usable energy in the form of ATP. Look up at me when you're done so I'll know. Okay, still some people writing? Let me move that out a little bit. Okay, so there's the equation. 6O2 plus C6H12O6, which is what?
Glucose.
Glucose. Yields 6CO2, carbon dioxide, plus 6H2O, water, plus ATP.